All right. Hello there. This is the walkthrough video for Monday. It is April 7th. There is no bell work this week because of exams and me needing to make sure I focus my energies on other things in the next little bit to help support you. I have some lab to prep. Um, However, I want you to think about one thing you needed to practice or review before this week's exam. Remember, the College Board suggests that this unit is 9 days long, and so our exam is going to be this Thursday. What about that thing you need to practice is tricky or challenging? Or like, do you know the content? You need to just finish your study folder or finish up a couple more Unit 5 notes. Just be able to think about that and maybe even make yourself a list for the next couple days. All right. So today we're going to work on predicting the likelihood of recombination between different genes in meiosis. And the reason is that this is why crossing over leads to so much variation. So we're going to talk about an assignment called recombination in linked genes. It is the last assignment other than the FRQ progress check, which we'll do tomorrow for this unit. And then I'll do a notebook check for you sometime soon. Those notes are last call due on Friday, May 2nd at 2:40 p.m. That's our new grading deadline. It is the Friday before the AP exam. All right, so combining these two ideas, because today we're going to think about what happens when genes are on different chromosomes and far apart on the same chromosomes. We know what can happen here, but crossing over means that there's a chance that stuff kind of gets remixed. Now, when they're very far apart from each other, kind of across the centromere in particular, these and these are really hard to tell apart because of the gametes that get made. But when we look at some other examples, like the ones in today's assignment, you're going to see that can be some big differences. So, for example, here are some that are really close together. So mostly you get the chromosome from genetic granddad and the chromosome from genetic grandma, but the gametes that get made, a very small fraction of them will have that recombination. So anyway, this assignment is based on and uses the Khan Academy webpage about linkage mapping. And it says here, remember that when genes are found on different chromosomes or far apart on the same chromosome, they sort independently and they're said to be unlinked. When genes are close together on the same chromosome, they're said to be linked. And that means that the alleles or gene versions that are already together on one chromosome will be inherited as a unit more frequently than not. We can see if two genes are linked and how tightly, how closely they are together by using data from genetic crosses to calculate recombination frequency. That's how likely, I don't know why there's not a parenthesis here, it's so irritating, how likely crossing over makes remixed chromosomes that mix up the two parental genes. By finding recombination frequencies for lots of gene pairs, we can make linkage maps that show the order and relative distances of the genes on the chromosome. This is genetics before there was DNA sequencing. And fruit flies were a really popular place to do that work. And so you'll see fruit flies are listed here. So together we're going to use data from genetic crosses to identify how closely together linked genes sit on the same chromosome. And so you're just going to visit the webpage and really use the text and do what it tells you to do and fill in the blanks and do that sort of thing. And then on the back here, we've got an example of a mapping pattern. And you can think about these questions, which would be great, but you definitely need to be able to get to here and be able to do that work. So, all right. And then I wanted to remind you, you can use chi-squared in this situation also in trying to figure out if genes are really closely linked together, what your expectation is in all of these situations, like this one and this one. Remember, you're going to use simple Mendelian dominance and recessive, even if you're looking at something and you're like, yeah, only 2% of those gametes are the recombinant phenotypes. Pretty sure there was recombination. You're always going to do chi-squared with the null hypothesis as simple Mendelian genetics. This is an FRQ, so you should pause the video and take a look at it. Okay, great. Did you do it? Thank you. And then I'm going to ask you, what do you know about this cross? So just looking at it, what can you decide about this cross? You don't have to do a chi-squared analysis, but just looking at it, what do you know about the alleles? What do you know about inheritance? Here's cross 2. Pause the video and do that same work. Cool. And then hopefully you were able to think about these. Will you pause the video and take a look? Okay, great. So now it's important to contrast that with this situation. Okay, standing here awkwardly. What do you know about it? Right? What can you tell about it? Pause the video. Yeah. So you can know that this is an example of some of this stuff, and this is why. So I'm not saying it out loud because I'm recording this during first period. All right. Now, hopefully you know these words. If you don't, You can use a lot of different resources to be able to do those. And if you haven't done the Unit 5 notes, I strongly suggest you take care of those. All right, that's it for me. I hope you're doing well out there. Take care out there and see you soon. Bye.